Hi everyone. This video is going to be an introduction to energy production. So the cells need energy to make proteins to do lots of different things, and this is going to serve as an introduction for that. This video is going to provide you a lot of context and introduce the concepts that we're going to be covering in depth in the next exam block. You'll also be doing work on these processes of photosynthesis and oxidative phosphorylation in your lab. So you'll really know these well by the time you're done with this. So let's get started. So energy production. The first thing we want to talk about is general energy production. So what does the cell do to make energy, right? Energy that it needs to do work. So it's going to take energy from outside of the cell. So energy from food. So food consumption in some way will make sugars, and that will go through the process of oxidative phosphorylation. And then pumps will move hydrogens across the membrane. Those hydrogens will flow across and make ATP. Or it might take energy from the sun. When it takes energy from the sun, the same thing is going to happen. That energy will be used to move hydrogens across a membrane, create a gradient, and then that gradient will flow back across the membrane and be used to make energy. So in both these cases, right, and this is the whole concept, photosynthesis and oxidative phosphorylation, so energy from light, energy from food, both do the same thing. They take energy from those sources and they create a gradient. So you can think about this if you like do something and you want to take a big rock and push it up a hill. That's creating a gradient. So you've trapped a lot of energy in that hill by pushing up the rock, and that's a whole big hill to roll down. So when you let the rock go, it rolls down on its own. That's what you're doing here. You're taking the energy, you're pushing the protons to make a gradient, and then when you let them, they're going to flow across the membrane naturally because you've made an area of high concentration. We're going to talk a lot about this and the detailed mechanisms for each of these. So again, this is the idea that these protons are what's moving here, right? So when you have something like a battery, the electrons are what moves around and is the energy. It's the same thing here, but what the electrons are used to do and what the energy from those high energy electrons do is make ATP. And that's what the cell uses as energy. It's not actually electrical energy. It's not the same things as what runs through your wires. But those electrons are the energy, and electrons do move in both of those processes. And we'll talk about when they move and where they move in those processes. So that is, electrons are involved, but energy can also be a stored concentration, right? So those protons that you put up in there and they flow across on their own. And that's the idea in the second point there of pumps moving molecules to store energy. When you move those hydrogens, you haven't stored electrons. It's not a battery, right? There's a battery here to give you this idea that like you can use energy to move water, right? You can put it up into a water tower and then it flows out and feeds the town. So that's what you're doing with the energy from the food and the energy from the light, which are the electrons, but you're not storing the electrons. What you're doing is making a gradient. You're moving a molecule to make a high concentration gradient, and that's where you store your energy. Okay? And these pumps work through conformational shifts. So we've talked about transmembrane proteins that have to flip around. And so the way that this one works is these proton pumps exist in several states. So one, the closed state all the way on the on the right over there. So they exist in this state. When they open up toward the matrix of the mitochondria, which is where all these protons are, they will have a high proton affinity. Then they close. When they open, they have a low affinity for protons, so the protons move out. Right, so again, when they're open towards the matrix, as you see in this diagram, which is the inner part of the mitochondria, they have a high affinity for protons. So when protons come in, they'll stick. That's what high affinity means. The protons are randomly floating around due to Brownian motion. And when they come in, they'll stick, and then it closes. When they open the other side, now this opening, this top movement, is the one that requires energy from electrons to happen. So electrons move, and that allows this to open up and have a conformational shift. And when it opens up, the protons are still there, but now there's a low affinity. So something about that conformational shift moves around some amino acids so that the interactions between all of these things are not as strong. And so the proton goes out. And then it goes back to this, and it flips around and goes back to normal and just keeps doing this motion a bunch of times. And so that's what that proton pump is doing. So the energy from the electrons is used right there in that little blue spot over there, right? That's it. That's where the energy is used. And so when we look at this potential energy, right? So what we've captured in terms of capturing those electrons and making a gradient, this potential energy in the form of this gradient and electron movement is going to be used to make ATP. So in both these cases, when you've got electron movements, you're going to make NAD+. When you have the electrons going from NADH and the electrons go on to oxygen and hydrogen and make water, that electron movement is going to power that conversion of ADP plus phosphate, inorganic phosphate, into ATP, which is the high energy molecule. So this conversion is electrical in terms of the electrons moving from NADH to form water. And that transfers it into chemical, adding that phosphate bond. That's a high energy bond. And so when you have that high energy bond, you now have ATP. ATP is the energy of the cell. Electrons are only used for this, right? To power this conversion of ADP and PI into ATP. And it does that using an electron carrier. You don't just have electrons flowing around the cell. What you have is you have electrons that are put onto a carrier molecule. A carrier molecule in that context, the previous slide, is NADH. That's an electron carrier. It's carrying the electron, moves it, and allows it then to go to water. And NADH and NAD+, they are just to carry electrons. And so what they're doing is something like this. So when you look at this molecule, they're like trucks. So they're going to, a carrier without an electron will come through, grab it off of food, which is an energetically favorable reaction, carry that energy over, and use it to power the production of new molecules in the cell by attaching that energy to it. And so when you break down food, that's catabolism. When you build up new molecules, that's anabolism. All right, so again, these carrier molecules are carrying the energy to these different places. So generally speaking, that's how the process works, right? To review, we start with energy coming in photosynthesis and oxidative phosphorylation. It pumps protons. Those protons form a high concentration gradient that then flows down, and that powers the production of ATP. This is very similar to how a battery might be used to move water, or electricity might be used to move water around, right? Say you're putting it into a water tower. You pump the water up, it hangs out in the water tower, and then gravity pushes it out. The pumps that do this for the protons are these pumps that undergo these conformational shifts, in which one conformation has a high affinity for protons, and so when they hit, they stick, and then when it shifts to the other one, they have a low affinity, and so when it hits that, they go out into the high concentration area. That shift requires energy. That's how it can put a molecule from an area of low to high concentration against the concentration gradient. The energy that's in there, right, that energy from electron transport, is accomplished using this, and we're going to get into the details, right? So that electrons in NADH get transferred onto oxygen and hydrogen to make water. That transfer powers the production of ATP. So that energy allows that ADP plus P to make a covalent bond and make ATP. And now you have that energy in the form of a high energy phosphate bond for ATP. How those electrons go and what NADH is NADH is a carrier. So it's like a truck. It picks up energy from food, carries it over to the other molecules, and allows it to be placed there. Okay? So how do we go from light to food, right? So this is photosynthesis. This is something you're going to be covering in your labs. We're going to talk about it in class as well. But generally covering photosynthesis. So there are two parts to photosynthesis. So when we talk about this, we have to talk about the things that happen in the light that are dependent upon light, and we have to talk about the things that are light independent. Okay, so things that need light to happen and things that can happen no matter how much, no matter whether there's light or not. So we've got light that can be made into ATP and electron carriers. That part requires light because that's where the energy comes from. But then you can take that ATP and electron carriers and you can use that to combine carbon dioxide into big long chain molecules like sugars and amino acids and fatty acids. And those are light independent reactions. And so what you need to remember is that white light is made up of all wavelengths. But chlorophyll only absorbs certain wavelengths. This is the absorbance spectra for chlorophyll. So you can see that they absorb blue light really well, and they absorb red light really well. They don't absorb green. Chlorophyll is why plants are green, because chlorophyll is green. An object is green because you're seeing the light that reflects off of it. So it's reflecting back the green light and absorbing everything else. So when you do this, right, I'm going to show you the next picture. We're going to talk. It's going to be for photosynthesis. I'm going to walk you through it. You're going to go in depth in this in your lab. We're going to talk about it in a lecture. So I'm just going to introduce this idea to you and point out the other places that we've already talked about just in this video. So if you look in B here, what you have is a summary of what happens in the chloroplast in photosynthesis. And so light will hit a photosystem. That light will be used to put energy onto an electron. That electron will go down to another photosystem. The energy from that electron will be used to move protons across a gradient and make that gradient. Then another photosystem, more light will hit there. The electron will go up and it'll be put onto a carrier. So you've moved the protons and you've made a carrier molecule. Those can then be used to do carbon fixation. And so then you get ATP, you get carbon fixation, you can take carbon dioxide, make all the covalent bonds between the C's, and then you can get carbohydrates. To me, right, this is the magical sugar fairy, because the chloroplast will take light and gas and make carbohydrates, make food, which is wild, right? It just takes gas from the air, strings them together in a long chain, and that's carbohydrates. So that's how we get from light to food. Now, how do we get to food, from food to energy? We don't have this, right? And plants don't use light as their power. They use light to make food, and then they consume the food. So they use sugar and carbohydrates the same way that we do. They just use light to make their own carbohydrates, whereas we have to consume other things. So how do we get from food to energy? The answer is cellular respiration, right? So we go from food and water, and we get ATP, NADH, carbon dioxide, food and oxygen, and we get ATP, electron carriers, carbon dioxide, and water. Okay? So the energy is ATP. We have what are called electron donors. These are the carriers. That's NADH. That's used to make more ATP. And then you can use that to make building blocks to make more molecules. Some of the food will be broken down using metabolitic processes to make amino acids and all the other things that we think about in the cell. And so this process is cellular respiration. So cellular respiration is this idea that you can take this food and break it down slowly and capture all the energy. There are kind of basically two ways to get energy out of a system. You can just break it down real fast all at once. In this case, you could burn sugar. That is going to release all of the energy. But it all comes out as heat, and none of it gets stored, right? And if you burned all the sugar that was in your body, while it's in your body, then you would be on fire, and that tends not to be good. So what the cells do is called cellular respiration. So they're capturing in a stepwise reaction, in an oxidation reaction, so adding oxygen to break apart bonds, you do small reactions that overcome the energy of activation, but in small ways, and you take off a little bit of energy each time and put them onto those carrier molecules. That captures the energy, and it can be used in other processes. Now, you always lose energy as heat. That's just how it works, and that's okay. But in this case, with the stepwise oxidation, with respiration, you can capture that energy and store it rather than just having it all go out in the form of heat. Right, so like I said, metabolism is going to take food and break it down. So you've got catabolic pathways, which are going to break things down. You always lose stuff at heat, but you can get building blocks, amino acids to build proteins, fat to build membranes, right? Lots of the things that the cell needs. You get those building blocks, and you get energy to do other processes. Part of what this class is learning about the other processes and how this works. So we'll get there. Like I said, this video is an introduction. And then you can have anabolic pathways where you use that energy and those building blocks to make the molecules the cell needs. Now, if you look, you may be like, well, why are you breaking it down and then making it? You're breaking certain things down. You're breaking down sugar, for example, and building new things out of that sugar, right? Or you're building down protein that you take in and putting it into molecules that the cell needs to work, not just the molecules that you consumed as food. So you're changing the shape of those things. And so there's a couple stages for food breakdown. We're going to talk about these a lot. This is a pretty simple modification, just a slight change from the book, but the figure's in there. So the first thing you have to do is consume the food. So you're going to break it down into digestible subunits, right? So you got to take the big piece of bread, chew it, have it go through your stomach, all those processes. If you're interested in that, anatomy and physiology is great. Break them down into sugars, amino acids, and fatty acids. So you break all these molecules down with digestion. Then you can take those subunits, sugars, amino acids, fatty acids, and you can process them into ATP and electron donors, and that's what's taking place in 2. So you're taking all those, you're making ATP, you're making electron carriers, and then in oxidative phosphorylation, you take all those electron carriers and make a whole bunch more ATP. And so this is that process, again, kind of very, very roughly as a summary in the mitochondria, right? And so again, you've got food-derived molecules. You're going to take electrons off of those using electron carriers. Those electron carriers are going to use those energized electrons, move protons across a gradient, and those electrons will go onto water, right? And those protons will then make ATP, right? So again, if you want to compare the two of them, this one is taking food, using the energy to pump protons, and then your final byproduct is water. Whereas if you look at photosynthesis, photosynthesis is taking water, applying energy to those electrons, and using it to make the food, right? So they are kind of reverse of each other. So let's talk about how we go from food to everything else, because this is an important part. We're not going to get into the details of this, right? This is a big focus of biochemistry. And so if you're really interested in how a cell might take these ingredients and make molecules with them, biochemistry, that's where you can learn all about that. But just kind of briefly go through some stuff. Metabolism, and when we're digesting these foods, you make more than ATP. I've said, you get these building blocks and you can make other things with them. So this is glycolysis in the citric acid cycle that we're going to learn about. And look at everything that comes off here. You've got a whole bunch of amino acids. You have heme molecules to carry oxygen in the blood. You've got chlorophyll. You've got cholesterol, fatty acids, purines, which become the nucleic acid, pyrimidines, lipids, right? So a whole bunch of this stuff, when you have glucose, sugar, your body will process it and make it into all of these other things. Now, again, you don't have to know this. I want to show it to you so that you are aware of the context of the stuff that we're going to be talking about. It can also get crazy. So this little pathway is highlighted in red here. This is all the other, and this is not even all of it. This is like each of those dots is the product of an enzymatic reaction to produce things in the cell. So it's crazy complicated, right? Again, I just want to show you this. So again, you see the context of like glucose is just a big part of this whole machinery in the cell to make the things the cell needs. And so through all of this process, as the cell is using energy, right, either from light or from food to make ATP, to make the molecules it needs, it's going to produce waste, right? And so fuel is going to come in through the cell membrane using a glucose transporter. It's going to do metabolism of food. It's going to make ATP. It's going to make all the stuff it needs from that food. And then it's going to make waste. And that waste has to go somewhere too. And that's excreted outside of the cell using exocytosis. And we'll talk about some ways that the cell can do that as well. So that's all we have for introducing the idea of using energy, right? And it's this introduction to how the cell can make energy, the types of things it might want to do. We're going to talk about many of these things in a lot more detail, but this video serves to kind of give you an introduction to that so that you can be aware when we're talking in class, when you're in your labs, where does this fit in the cell? Okay, so I'll see you all in the next video.